Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're very happy you're here today. And we'll be talking about active learning and drug discovery today with Pat Walters. Hi, Pat. How's it going
Hi,
this morning?
hi, Dennis. Thank you for the opportunity to speak today.
Yeah, no, we're really glad you're here today. We're thrilled to have you. So it's such an honor that you're here today. And now, without further ado, I want to introduce our guest, Pat Walters. So, Pat is the Chief Data Officer at Relay Therapeutics in Cambridge, Massachusetts. And prior to joining Relay, he spent more than 20 years at Vertex, where he was the global head of modeling and informatics. And Pat is also the 2023 recipient of the Hermann Skolnick Award for Chemical Information Science from the ACS. He is a member of the editorial advisory board for the Journal of Chemical Information and Modeling and Artificial Intelligence in Life Sciences, and previously held a similar role in the Journal of Medicinal Chemistry. Pat is also the co-author of the book Deep Learning for the Life Sciences, published in 2019 by O'Reilly and Associates. On top of that, Pat has numerous publications and patents that he is a author or co-author of. Pat is also known for his blog, Practical Chem Informatics, and several useful tutorials that he puts out on GitHub. So he's doing a lot of amazing service for the community. He received his Ph.D. in organic chemistry from the University of Arizona, where he studied the application and of artificial intelligence in conformational analysis. And prior to obtaining his Ph.D., Pat worked at Varian Instruments as both a chemist and a software developer. Prior to that, he received his bachelor's in chemistry from the University of California in Santa Barbara with this beautiful campus right on the ocean. So in the good old Flash Talk tradition, we wanted to start with a few warm-up questions for Pat. So Pat, again, welcome to the show today. We'd love to hear a little bit more why you started a career in drug discovery. What inspired you to do so?
Yeah, I think I started out as more of an algorithm guy. I really liked writing software. I loved chemistry, and the chance to put those two together was very inspiring to me. And then, after a few years at Vertex, I spent time and started working directly on drug discovery projects. And then through my career at Vertex, I got to see the impact that some of the compounds that we had come up with had on patients. And, I ended up getting to meet a lot of the cystic fibrosis patients at Vertex and hearing their stories and hearing how the influence of how what we had transformed their lives was incredibly inspiring for me.
Wow, yeah, that's such a great story. Yeah, that's always very touching to hear stories from patients and then how you can actually, with your day-to-day work, impact their lives. So very inspiring.
It's the best thing in the world.
Totally. I think we're very fortunate to work in this industry.
I agree. I feel very lucky.
Yeah. And yeah, maybe elaborate a little bit on your favorite molecule, Pat. What's your favorite molecule and why?
Yeah, my favorite molecule right now is VX548, the NAV 1.8 blocker that was approved last week by the FDA. So this is a compound that was developed at Vertex. A lot of that work went on during the time I was there. So I kind of had a front row seat watching the development of that compound. It's a novel mechanism. It's, has the potential to have a huge impact on patients' lives. And when you look at it, it's just a beautiful molecule. I think someone commented yesterday on the internet that that molecule is like a Mozart symphony where every note is in the right place.
Totally. Yeah, I like that molecule as well. That's it's also like the time it comes for the opioid epidemic that's going on in the US. It couldn't be a better timing. So really glad to hear you played a role in the discovery of this molecule. So
Well,
congratulations to you
I did not play a role in the discovery of this molecule. It was developed at Vertex while I
while
was
you
there.
were there.
I was not part of that program.
Awesome, awesome. Yeah, and what are you excited about in drug discovery today in general, Pat?
I'm very encouraged by all the work that's going on with machine learning. I think there's still a lot of hype, but there's substance among that. And I think it's enabling us to evaluate more ideas, putting these tools in the hands of drug discovery teams, I think is enabling people to make better decisions and evaluate more possibilities.
Awesome. Yeah, and we're looking forward to hear more about that today in your talk as well. And maybe if you could go back in your career and give yourself some career advice, what would you tell your yourself? And maybe in a situation today, what would you tell a scientist who's just starting out their career in drug discovery?
I think try to be as well-rounded as you possibly can. If I had it all to do over again, I would probably have learned more biology. I've ended up learning a lot of biology on the job, but I think the people who are the most effective in drug discovery are the people who are well-rounded, the people who are chemists who have a strong appreciation for biology and a strong appreciation for chemistry, or pharmacologists who can think about how to optimize a molecule. So, to me, the biggest thing is trying to be as well-rounded as possible.
Awesome. Yeah, no, that's I think such a great answer. Like, I mean, continuous learning. It speaks for like continuous learning of the whole field of drug discovery. And again, it's so many disciplines that you highlighted that participate in drug discovery and you really make a molecule come through and ultimately be a medicine.
Yeah, I mean, the best thing about this stuff is so much fun. And it's great being able to now, go look at Drug Hunter and say, oh, hey, I didn't know anything about that program. Maybe I should go read this paper.
Totally agree with that. Well, with that, you like read my mind. What do you most like about Drug Hunter?
I like two things. One is these molecule one pagers because it gives me a way to get a quick view into often therapeutic areas and targets that I don't work on, but where there are ideas that we can perhaps borrow. And then also the patent alerts. I love looking at those just to see the sorts of molecules that people are making. And again, this can give me inspiration. I can see that and say, oh, I hadn't thought about this particular bit of functionality. Maybe I should go read more about that. So it provides me with a nice entry into the literature and kind of helps to direct a lot of my reading.
Awesome. Yeah, thank you so much, Pat. So before we launch into your presentation, I want to quickly show you those patent highlights to see. We actually just published. We just published the December 2024 highlights, patent highlights. So it's in a Gazette style, comes out every month, and the team is currently working on the highlights for the month of January 2025. So let's click into those, double click into those and see what we have here. So we analyze the entire patent landscape here. Every Thursday, as you know, patents will drop in WIPO, and we will make a list of the 200 to 300 most exciting patents and highlight approximately 6 to 10 areas where basically really cool developments have happened. And what do we do then? So here, as you can see, we're talking about PPI inhibitors. So we extracted some structure here from a patent here from Innerline Therapeutics. You can see they highlighted 6 different chemical series as those RIPK2 XIA PPI inhibitors. We compare those with a patent that was filed at a similar time here by Odyssey. And with some compounds that are a little bit more advanced, compounds that have been tried in clinical trials, and even with different kind of MOAs, different modalities. We talk a little bit about the target. Why is the target important? What is the competitive landscape? What are some of the potential indications for this particular MOA? And then we kind of talk a little bit about, the prospects of this molecule, how it differentiates. And we do this again for a variety of different areas. So you can see data from those patents. You can also oftentimes see curious functional groups that, Pat pointed out. I had never seen this motive here before in a drug or a drug candidate. So we kind of try to surface those really interesting looking structures that you can basically use for your own programs. Again, we compare with other chemical matter that's out there. So, for example, here was a patent for the orexin 2 receptor agonists by Lundbeck and for narcolepsy. So we compare these chemical structures here with a patent that was filed by Vertex in the same space and a patent that was filed by Alchemir, so that you can see how structurally different those compounds are. We also do point out, things that you should be aware of in patents. So, for example, here with Carmon Therapeutics, USB28 inhibitors, that's an interesting target, first of all, for obesity these days. So everybody's, of course, working on GLP-1 receptor agonists. But yeah, Carmon Therapeutics doesn't have USP28 on their website, so it's of course a new target in that space. But it's also very well known that USP28 inhibitors might potentially hit USP25, which is very closely related to USP28. So we point out that in this patent, there's no data available on USP25, but we do highlight a few compounds from the literature where USP28 and USP25 data is available, so that you can kind of see how they structurally differentiate. If you're interested in learning more, just let us know. Feel free to contact us. And with that, I'll stop sharing and hand over the floor to Pat for his talk on active learning in drug discovery. Really excited to hear more from you, Pat.
All right, are my slides showing?
Perfect. Yeah, I can see them in presentation mode.
All right. Thank you, Dennis. Thank you, Drug Hunter team, for the opportunity to give this flash talk. I want to tell you about some of the work that we've been doing with active learning. And I'm going to tell you two different stories today with a common theme. And the idea is that we've got a lot of computational workflows, but these workflows can be computationally expensive and actually cost a lot to run. So we've got techniques like free energy perturbation, which we can apply on small sets of compounds, but often are difficult when we get to larger sets. And then I want to tell you another story about ultra high throughput virtual screening, where there are now very large libraries in billions and even trillions available. And how can we, in a computationally efficient manner, process libraries like those? So we'll start out talking about free energy perturbation. And free energy perturbation is a very powerful computational technique that's incredibly useful in a lead optimization setting. So many times in a lead optimization setting, you have a molecule and you want to either increase its potency or perhaps maintain potency while altering other properties and doing things like optimizing pharmacokinetics. So the way that free energy perturbation works is we start with one molecule. We do a molecular dynamics simulation, and then over the course of that simulation, we morph the molecule into another molecule that we're thinking about making. Over the course of that, we can get an idea of the difference in the relative free energies of binding between the two molecules. Now, these calculations are very accurate, very useful, but they are time consuming. So typically, on a state-of-the-art GPU, it's going to take you somewhere between 4 and 8 hours to run these free energy calculations. So this is fine if I have tens of molecules that I want to evaluate. I might even be able to start looking at 100 molecules, but I'm not going to be able to process very large data sets where I have thousands of molecules. And simply by doing enumerations over known chemistry, we can often get into cases where we'll have 10,000 or more ideas for molecules that we could potentially make. So how can we process those larger libraries with these more computationally expensive techniques? So let's just take a look at an example here. So what we're looking at in this plot is 10,000 molecules. So these are actually real molecules that were taken from a BMS TIC2 patent. So what we're looking at is what's called truncated stochastic neighbor embedding. What this is a plot of the chemical space. Now, many of us look at this as scientists and say, okay, what's the x-axis? What's the y-axis on this plot? I'd like you to look at this as a different way and think about this as a map and with north, south, east, west. So, and on this map, each point represents a chemical structure. Structures that are close together are chemically similar. Structures that are farther apart are more chemically distinct. Now, given this and given this set of 10,000 molecules, we probably wouldn't be able to run free energy calculations on every molecule. However, what if we could build a machine learning model that would predict the output of the free energy calculation? And one of the nice things about machine learning is that it's very fast. So I can train a machine learning model and do inference with that model on a new set of molecules. If I have 10,000 molecules, it's going to take me about 7 seconds to run. So because machine learning is millions of times faster than these free energy calculations, we can take advantage of that and come up with a hybrid approach where we run a free energy calculation, use the results of that free energy calculation to train a machine learning model, and then run an iterative cycle where we run free energy calculations and then use those to train a machine learning model. So one way to look at that is that we can build this machine learning model, which will predict the delta G, the binding free energy of a molecule bound in a particular receptor as a function of a set of molecular descriptors. So we've got a set of molecular descriptors that we can calculate from a chemical structure. We can use that to build a predictive model. So what we're going to do is we're going to start out and we're going to just select 50 molecules out of those 10,000. We're going to use those 50 molecules then to build a machine learning model, use that machine learning model to predict on the remaining 9,950 molecules, then take the 50 best predicted molecules from the machine learning model and run another free energy calculation. And we can just repeat that process through a few cycles, and I'll show you how we can rapidly identify the best molecules out of these large data sets. So let's just start out with that same example with 10,000 molecules from this BMS TIC2 patent. So we're going to start out and we're going to randomly select 50 molecules out of that set. So then we'll take those 50 molecules and we'll run a free energy calculation with those. So now I have 50 chemical structures and I have 50 results from a free energy calculation. So I can then take those 50 chemical structures, generate a set of molecular descriptors from those 50 molecules, and build a machine learning model that relates those molecular descriptors to the free energies calculated in the free energy perturbation calculation. So here's the result after we've done that first round. So we've now screened only 0.5%. So we've screened 50 molecules. You'll see in the plot over on the left, you see the molecules that we selected. In red, you can see that those are randomly selected over chemical space. In the plot on the right, we're looking at the free energies of those molecules from an FEP calculation. You can see here what we have is, as we would expect, a normal distribution centered close to zero. Now, the other thing to remember about these is a more negative value in the FEP calculation, a more negative delta G indicates a tighter binding molecule. So what we're going to do now is we'll take those 50 molecules and the 50 results from the free energy calculation, we'll build a machine learning model, and then we'll use that machine learning model to predict the free energies for the remaining 9,950 molecules. We'll take that prediction, select the 50 best, and run another free energy calculation. And that's what we're looking at here. So now, if we look at the plot over on the left, we'll see in dark blue the molecules that we selected in the previous cycle. In red, you'll see the molecules that we selected in this cycle. So we've only sampled 100 molecules. We've sampled 1% of the total. Over on the right now, in red, you see the distribution of free energies from this particular iteration. And in blue, you see the free energies from the previous iteration. So this is nice. We've already shifted that distribution significantly to the left, and we're identifying molecules that are predicted by the free energy calculation to be tighter binders. So we're going to repeat this again. Now we have 100 molecules. So we use those 100 molecules to train a machine learning model. We'll then use the selections from that machine learning model to select another 50 molecules and run another 50 free energy calculations. And we did that. You'll see now here's the result from the next cycle. Again, in red are the molecules that we selected in the last round. In blue are the molecules that we selected in the previous round. And if you look, you'll already see that the model is starting to focus in on certain areas of chemical space, which are shown in red. On the right, we look at the distributions of free energies, and we'll see that, again, we have a slight left shift in that distribution in the red curve, which represents the current round. So now we've sampled 1.5% of the total. We now have 150 molecules. So what we'll do is take that set, build another machine learning model, and select another 50 molecules. As we do that, you'll see that again, now here's the current round again in red. You see it focusing even more on certain areas of chemical space. When we look at the plot of the curves over on the right, you'll see that again, that distribution has shifted to the left toward a more negative delta G, and we're finding molecules that are going to be tighter binders. So now what we've done is we've only sampled 3%. So we've sampled 300 out of 10,000 molecules. Now, what the question I want to ask is, by sampling 300 molecules, how many of the best molecules did I find? How many of the top 100? Now, the part of this that I haven't told you yet is we did this project as a collaboration with colleagues at Google. One of the great things about working with people at Google is that they have more or less an unlimited number of GPUs. So together with our colleagues at Google, we were able to get the ground truth. We ran free energy calculations on all 10,000 of these molecules. So we know what the best 100 are, and we can see now how many of the best 100 our active learning protocol identified. And so if we look at the plot over on the left, what we see in red now are the molecules from the top 100 that we found. In gray are the molecules from the top 100 that we didn't find. So you can see that the model focused on certain areas of chemical space, and there are areas that we didn't explore, and there are molecules from the top 100 that we missed. However, if we look over at the plot on the right, you can see that by only sampling 3% of the 10,000 molecules, we were able to find 49 out of the top 100 molecules. The other thing we wanted to look at was how much chemical diversity were we covering? So we took those top 100 molecules and clustered them. When we cluster those, we find that there are 35 clusters in there, and we were able to identify 19 out of those top 35 clusters. So by searching a relatively small number of compounds, really only 3%, we were able to identify half of the top 100 molecules. So this work we published at the end of December 2022 in Artificial Intelligence in the Life Sciences. This work is published now, and the code is available. So if anyone wants to use it, this code is available on GitHub. So I'm going to shift gears just a little bit, and I'm going to tell you about another story and show you how we've been able to apply a different variation on active learning into virtual screening and enabling ourselves to virtually screen very large libraries. So I want to tell you a story about how we've been able to apply this another active learning technique, Thompson sampling, to virtual screening using protein ligand docking. So I'm sure many people in the audience are very familiar with protein ligand docking. This is an efficient way to perform virtual screens. So if you have a protein crystal structure, you can dock small molecules into the protein binding site. Many software packages will allow you to do this flexibly and explore conformational flexibility. So protein ligand docking is an incredibly powerful technique, but it does have some limitations. And a lot of those limitations have come about due to advances in synthesis on demand chemistry. So up until a few years ago, there were a couple million molecules commercially available, and we were able to more or less through brute force virtually screen everything that was commercially available. But now we've got libraries like the Explorer collection from eMolecules. It's more than 1 trillion. There's Wushi's Galaxy library, which is tens of billions. There's the enamine real space, which is constantly growing. And it has now reached a point where we really can't brute force this problem anymore. And just to highlight this, I can go back to 2019, and this is a slide from a talk that my colleague Levi Pierce presented at the AWS reInvent conference. So at that time, we were very proud of ourselves because we were able to put together a workflow on Amazon Web Services that enabled us to spin up 100,000 CPUs, and we could dock 1 billion molecules, which was the size of the enamine real collection in 2019. So we could brute force dock 1 billion molecules in a day. That would cost about $12,000 in compute, which is reasonable. But now the game is no longer 1 billion molecules. We've now reached the point where there are hundreds of billions of molecules available for purchase for synthesis on demand. So, it's fine if I could say, great, I'm going to dock 1 billion molecules for $12,000. But if the game then becomes 50 billion molecules, and instead of costing $12,000, that costs $500,000, I could do that exactly once, and then I would no longer have a job. So we need more efficient ways of approaching these ultra large virtual screens. And I want to tell you about one technique that we've been taking advantage of. And this is not a new technique. So I'm going to tell you about a technique that we use called Thomson sampling. This was originally published by William Thomson in 1933. And this technique and variants on it, and the technique has been rediscovered several times, but it's been used most widely as a solution to what's called the multi-arm bandit problem. So a lot of great statistical work came out of people who wanted to win at gambling. And one technique that's been widely applied to this multi-arm bandit problem are techniques like Thomson sampling. So I want to demonstrate this to you with an example where we're going to take a look at slot machines. So imagine a case where you've got 3 slot machines. I don't know anything about these slot machines or their propensity to pay off initially. So what I have is a uniform distribution associated with each of my slot machines. So what I'm going to do first is generate a random number from each of these 3 uniform distributions. And you'll see that in red in the center, I have the highest random number selected from the distribution. So what I'm going to do is I'm going to pull the lever on that machine, and you'll see that I won. So now I know that this machine in the middle has some probability of paying off. So what I'm going to do is adjust the probability associated with that machine. Now, given those modified probabilities, what I'm going to do is generate another random number. And again, I'll pull the lever on the slot machine that has the largest random number, which is the one in the middle again. I pull that lever. This time I lost. So now I've altered my distribution again so that I have more of a normal distribution associated with the machine in the middle. And I can continue this procedure where I generate a random number out of the distributions associated with each of the 3 machines. And then I select the machine with the highest random number. I pull the lever for that machine, and I adjust the distribution. And I can continue this process of generate a random number, select the machine with the highest random number, pull the lever on that machine, and adjust the distribution based on a win or a loss. So you can see that I'm keeping up this process. I continue to adjust these distributions. You can see that I'm actually doing pretty well on my gambling exercise here. Now you'll see we selected the machine on the right. We won with that machine. We'll adjust the distribution again, and we can continue this process. Now, you may be asking yourselves, okay, why are you telling me about slot machines, and how can we take some of these ideas and apply them to virtual screening? So let's take a look at that. And how would we apply that? So first of all, let's think about these large or ultra large chemical libraries. Now, when you look at these libraries, you have a large number of reaction products, but this can actually be turned into a smaller set of reagents. So if you think about this library here of amides, I've got 9 reaction products, but I can make that from 3 carboxylic acids at R1 on the vertical axis and 3 primary amines at R2 on the horizontal axis. So if we extend that idea to larger sets, if I have a 2-component reaction and I have 1,000 R1s and 1,000 R2s, I can get to 1 million products. If I do that with a 3-component library, and I have 1,000 of each reagent, I can get to 1 billion products. So can we address this search problem in reagent space, which is much smaller than product space? And yes, we can. So what we're going to do is we're going to treat each of our reagents as one of these one-armed bandits. So we could start out by just randomly selecting a pair of reagents. So we randomly select an acid, we randomly select an amine, we can combine those through a virtual chemical reaction and generate a reaction product. Now we want to take a look at docking. So the first thing we're going to have to do is then take that virtual reaction product and generate an ensemble of conformers for that molecule. I can then take that ensemble of conformers and I can dock it into a protein binding site. I can then take the score associated with the best scoring conformer, and I can propagate that back to the molecule and then propagate it back to my reagents, my chemical building blocks. So now you can see that in the same way that we were adjusting distributions based on wins and losses with slot machines, we can adjust the distributions associated with our reagents based on docking scores, and we can use those then to direct our search through the reagents for these very large libraries. And I want to show you now a couple of examples that demonstrate how well this works. So what I'm showing on this plot is a set of docking scores for the best 100 molecules from a library of 335 million compounds. So what we did was we use Thompson sampling, and we only searched 1% of these 335 million molecules. So through that search, we identified a set of molecules. The top 100 scores with a larger number being better are shown in orange. So in orange, I did 10 Thompson sampling runs. Now, over on the right, I told you before that we had done a number of large brute force virtual screens. So in green on the right, I have my reference scores. So this is the ground truth. I did all the docking and identified the top 100. In purple here, I took the 10 Thompson sampling runs in orange, concatenated them, and then took the best 100 unique molecules out of that. So you can see, first of all, if you just look across this, you can see that the score distributions look very similar, but we can dig in a little more and we can see how many of the top 100 molecules we identified. And you can see here, at the low end, we identified 89 of the top 100. At the high end, we identified 91. You can see in purple, the concatenation strategy didn't really help us. But by only searching 1% of the library using Thompson sampling, we were able to typically identify at least 90% of the best molecules. Now, one of the great things about Thompson sampling is that it's a very generalizable technique, and we don't have to apply that to docking. We can also use that for things like 3D shape searching. So I want to show you another example where we applied Thompson sampling using the ROCS method from OpenEye Scientific Software that searches for molecules with similar shapes and sets of pharmacophoric features. So the plot I'm showing you here is very similar to the plot I showed you for the docking. However, in this case, we only searched 1/10 or 0.1% of a large set of 234 million molecules. Again, we want to take a look at how many of the top 100 molecules we were able to identify using Thomson sampling. So again, over on the right in green, I have the ground truth. So we did the brute force search. Those are the scores of the top 100. Again, a larger value is better. In orange, you can see 10 Thomson sampling runs. You can see that those were relatively consistent. And then in purple, we have the concatenation of those 10 Thomson sampling runs. So as before, we can take a look at how many of the top 100 molecules we identified, and you can see that here again, we're hitting at least 90%. So at the low end, we had 91 molecules. At the high end, we had 95. You know, the concatenation gave us 96. So again, it didn't help us that much, but hopefully I've convinced you that this is a very efficient technique, can be applied, you know, whether you're doing docking, whether you're doing a 3D shape search, you can use a machine learning model. Thomson sampling can be coupled with a wide range of computational techniques. Now, this is all great, and I showed you a lot of retrospective results, but you're probably asking yourself, that's great, how well does this work in practice? And does it work prospectively? So I want to show you that. I just wanted to mention, we did publish this work at the end of in the middle of last year. The code for this is available on GitHub. So if you'd like to try it, please download that code. But I'd like to finish by talking about a real practical example. So this is some work that started during the pandemic. And this is collaborative work that we did together with Jamie Fraser's group at UCSF, Brian Schoiket's group at UCSF, and Frank Van Delft's group at the Diamond Light Source in the UK. So one of the promising targets for coronavirus is the SARS-CoV-2 macrodomain. And in a heroic effort, the Fraser group and Van Delft groups solved 235 different structures of fragments bound to the SARS-CoV-2 macrodomain. And this is great because it's an incredibly high resolution data set. I think the median resolution is just slightly above an angstrom. So we've been collaborating with Jamie Fraser since the early days of Relay. He comes to visit about once a quarter. He and I were sitting together. He was showing me this data set and said, well, what would you do with this data set? And if we think about the way that people typically approach fragment screens, there's one of two approaches. The first is fragment growing, where you have a crystal structure of a fragment, you look at different vectors coming off of hydrogens or places that you could make chemical bonds, and you can think about ways to grow that molecule into another pocket and improve its potency. You know, the other approach, which tends to be the dream, which is often not realized, is fragment linking, where you have two fragments, you try to come up with some chemistry to link those, and then hopefully you would get some multiplicative increase in binding potency. The problem is that that rarely works, because when you make that linkage, typically something shifts by a couple tenths of an angstrom and you lose a lot of binding affinity. With either of these approaches, you're typically looking at doing a lot of chemistry, and there's going to be a lot of experimental effort. So we were trying to look at a different approach. So we developed an approach, and other people have developed similar approaches, but I think ours has a cooler name. So we call our approach Frankenrocks. And Frankenrocks is really a conceptually simple idea. So we start with a pair of fragments. We then use that pair of fragments to do a shape search for molecules that have similar shape and pharmacophoric features. In doing that, we can identify commercially available compounds. So rather than spending months and large amounts of money on chemical synthesis, we're identifying molecules that we could purchase, hopefully for, on the order of $100 a piece. Now, there's a couple of things we had to consider here because we had 235 fragments. So that's 235 squared combinations. How do we figure out which pairs of fragments to look at? So we started with a simple distance heuristic and only considered fragment pairs where the distance between the centers was less than 20 angstroms. Now, there are also going to be pairs of fragments that aren't particularly interesting because they're like cases 1 and 2 over on the right where these fragments are sitting on top of one another. So we also just did a simple volume overlap calculation so that we could identify things like number 3 and number 4, where there was a chance that we could find a single molecule that exploited the interactions of both molecules in that pair. That was still a lot of queries. So that generated almost 5,000 queries. We used some additional heuristics and some visualization and pruned that down to 97 queries. We use those 97 queries with Thompson sampling to search the 23 billion molecules in the enamine real collection, which was the size of the database when we did this work. We then ended up with a set of scores. You can see that distribution of scores here. We picked then some of the highest scoring molecules, did a bit of just interactive visualization, and selected 32 molecules for synthesis on demand. From those 32 molecules, we were able to identify a number of molecules that then gave us crystal structures. So it's always great when you do a conformational prediction and then you get experimental validation. So you can see here on the left, I've got the original pair of fragments. In the center, I've got what we predicted from our Frankenrox calculation. And then on the right, we have the X-ray structure. We put those together and you can see that what we predicted is very, very close to what we ended up with structurally. So this is very satisfying. In addition, we were able to find a number of novel binders. So we were able to get crystal structures of 6 of those 32 hits. When those were evaluated in an HTRF assay, we were able to go from millimolar fragments down to molecules that had double-digit micromolar to triple-digit micromolar potency. Those were able to be progressed through through a couple rounds of chemistry down to molecules that were synthesized by our colleagues at UCSF, and there are now single-digit micromolar leads that have come out of this effort. This is published on arXiv. It's been submitted for publication. Hopefully it will be available very soon. I'm going to skip over this and just finish here. I think I've shown you a number of computational techniques and creative ways that we can apply computation, but I still think that this is only one part of the drug discovery process. And I think the best way to apply computation in drug discovery is in very close collaboration with experimentalists. So I want to thank all of my colleagues at Relay Therapeutics who worked on this, as well as our collaborators at Google and UCSF. I want to thank all of you for your attention. This is my website. It has links off to the papers, the tutorials, some of my blog posts, and the Git repos with some of this. So thanks a lot. Thanks, Dennis.
Thank you so much, Pat. This was an amazing talk. I mean, we have so many positive audience reactions. You're probably seeing this right now on your screen, and
I
uh,
love it.
yeah, I love it too. And we have 25 questions already in the chat. We're probably not going to be able to get through all of those, but you know, Pat, thank you so much. Let's maybe dive right into the questions, and I'll
Sure.
just start with the questions that got the most likes.
Okay.
So for the first project that you described with the FEP calculations, which molecular descriptors do you recommend for the use for this particular task?
In this particular case, we just used a straight Morgan fingerprint from the RD kit. I think one of the nice things about this is we were predicting a computed endpoint. And one of the things I find is predicting computed endpoints is relatively easy because there's not a lot of noise there like we have when we're dealing with experimental data. So this was just relatively simple. There's a tutorial in my practical cheminformatics tutorials that
Nice.
walks through the exact process that we did here.
Awesome. Yeah, that's super helpful. Yeah, and thank you for pointing out that all these models are available for people to try. That's really amazing. Um, Pablo is wondering, how would you balance exploitation, that means selecting the compounds with highest potency, while ensuring a consistent exploration of the chemical space to not miss any unexplored, uncertain areas that may contain hits um, out of the applicability domain of the ML models?
Yeah, and this is one of the things, and I have to make a confession here. We tried to be incredibly clever with this. One of the things I loved about the active learning project that we did was we did this with Google, and I think this was the first time that anyone had been able to get the ground truth for a large data set like this. And I wanted to evaluate a number of strategies for balancing exploration and exploitation because I'd seen other papers where people were saying, oh, I'm going to do 2 rounds of exploration, followed up by a round of exploitation, followed up by 2 rounds of exploration. And there was no real reason for any of this. It just felt good. So if you look at our paper, we evaluated 5 or 6 different strategies for balancing exploration and exploitation. We tried to be really clever. It doesn't matter. We found that just a simple, greedy approach worked incredibly well here.
Awesome, awesome. That's actually what somebody commented also in the chat. So, yeah, I mean, the audience captured that from your talk already. Kirill is wondering, is it possible that a set of molecules from a real medchem campaign in an FEP or ML example is biased compared to a completely unknown chemical space you would face in a new campaign?
Yeah, I think you do have limits when you're trying to go outside the applicability domain of a model. And one of the things that I like about this technique, and really both the active learning and the free energy perturbation work well within a chemical series. I think a lot of the things that we've shown here, especially as far as the active learning and the free energy perturbation, these are methods that are designed to work well within a series. If you try to take this, it's not going to work with an arbitrary set of molecules. Similarly with Thompson sampling. Thompson sampling is working off of a specific set of reactions. And you can't just say, I'm going to take Thompson sampling and apply it to some arbitrary set of molecules. So there are limitations here, but I think if you work within those limitations, the techniques can still be very powerful.
Sounds good. And VJ is wondering if the approach is appropriate for active cliffs as well.
Activity cliffs.
Activity clips, sorry, yeah.
Yeah, I think, it's possible that activity cliffs are going to trip you up. Those trip you up, regardless of what you're doing when you're building machine learning models. I think, especially though, in the case of free energy perturbation, you may be able to employ the physics to help you to, at least in some sense, avoid the activity cliff.
Sounds good. And then let's see what the next question would be. Oh yeah, people were asking what open source solution would you use for the FEP calculations? Do you,
There are several open source FEP implementations. So there's an open source implementation from the Open Force Field Consortium. There is an Amber implementation. So there are a few implementations out there.
Sounds good. Yeah, thank you. And then Antonio is wondering, I found that standard normalization techniques frequently result in severe information loss when used on typical physchem descriptors. Do you have any recommendations for normalization techniques on such data?
Yeah, I think it depends, at least in my experience, it depends on what the machine learning technique is that you're applying. I've found that sort of standard normalization techniques with things like message passing neural networks tend to work relatively well. There are other techniques. So if you're using something like a random forest model, or if you're using something, like gradient boosting machines, et cetera, tree-based models, you don't really have to do normalization. I see people doing things like normalization and feature selection with tree-based models, which is not necessary.
Got it. Yeah. Sebastian is wondering, given some binding events have specific requirements like halogen bonding or pi stacking, do you ever select in a partially biased manner to ensure that the ML algorithm do not ignore those interactions?
Yeah, absolutely. And then, the other thing that really comes into play there, especially when you're doing free energy, something like free energy perturbation, is the molecular mechanics force field that you're using and making sure that your force field has the appropriate terms in it to capture the interactions that you want to go after.
Yep, that makes
But
a
yeah,
lot of
I mean,
yeah.
you can use a variety of heuristics or tuning to push your search toward where you think you want to go.
Yeah, sounds good. And then Ilya is wondering about the Tanimoto score that you used. Ilya noticed that there's a value that's greater than 1, and just wondering what that Tanimoto score was.
Oh, that is what's called the Tanimoto combo score from the ROCS program from OpenEye Scientific Software. So there are two components to that. There's a shape component, and then there's what they call a color component that captures the pharmacophoric overlap. So each of those ranges between 0 and 1. So a perfect score for that Tanimoto combo is actually 2.
Oh, got it. Got it. Cool.
But
Um, and then
very, very keen eye.
oh, keen eye, the audience here. Like we're very detail oriented
Yeah.
here. Let's see. I think the next question is from Ravi. Could you elaborate on the machine learning methods used to train the models? Additionally, was 6 training cycles sufficient to achieve the desired accuracy in compound predictions?
Yeah, and so a lot of this is detailed in our active learning paper, but
Hmm.
in this particular case, we use Gaussian process regression. One of the things that I like about Gaussian process regression is that in addition to getting a prediction, you get an uncertainty associated with that prediction. So that gives you the ability to incorporate that uncertainty into your active learning process.
Yeah. And yeah, maybe I guess we still have more than 20 questions. Actually, we're not going to get through them today, but I think we'll end it here with one question from François Béranger. Uh, won't the FEP results be influenced by the predicted binding mode for those molecules? For example, you compute the FEP assuming a protein ligand, the protein ligand is placed correctly for each molecule.
Absolutely. And to be honest, I would not run an FEP calculation if I were not confident in the binding pose. We put a lot of effort into making sure that the molecules are correctly posed. This sort of comes back to what I was saying before. The best computational work is computational work that's done in close collaboration with experimentation. So, it's great when you have structural biologists who can provide you with crystal structures that you can then use to bias the poses that are going into your free energy calculation.
Yeah, and you showed that very beautifully in the talk, how you overlaid all of this and essentially got very similar results, which is always what's highly desirable for the practitioner.
Yeah, always makes me very happy.
Me too. Me too. If a prediction, is good, then and you can feel confident to use that model, that's makes me very happy as a medicinal chemist, so to say. All right, with that, thank you so much for an amazing, really incredible talk, Pat. Again, the audience really enjoyed this. Again, we had over 30 questions. We had more than 400 attendees here at the top. So it's really an amazing presentation, Pat. I thank you really for showing us and, really demonstrating the work that you and your teams did. Absolutely outstanding. I thank the audience really for the curious, insightful, and keen eye question, as Pat pointed out. And yeah, I would encourage you to dial in next week. We have a flash talk coming up with Gavin Hurst. We're going to be talking about Beyond the Molecule, Gavin's experience. He's been the CSO at Atomwise most recently. So we're going to talk a lot about artificial intelligence and just Gavin's perspective on drug discovery today. And with that, we close for today. And again, thank you, Pat. Thanks to the audience. And I wish everybody a wonderful day. All right. Bye bye. Thank you, Pat.